Hey all, so today we're looking at D2.2, which is gene expression. And the questions we're trying to think about is how is a gene expressed? How is gene expression changed in a cell? And how can a pattern of gene expression be conserved or maintained through inheritance? So we're looking at pages 636 to 647. This is HL content, and we're thinking about continuity and change. So firstly, gene expression, this is a pretty crazy diagram here. And what we're thinking about, genotype is all of the genetic information of an organism, while the phenotype is their total physical characteristics. Gene expression is controlled by transcription, translation, and protein function. And then genes are not only turned on and off, but they're up and down regulated. So that means they can do more or they can do less, or they can be turned on or they can be turned off. And the pattern of gene expression regulates how a cell differentiates. So production of messenger RNA is controlled by a promoter on DNA at the Tata box. So you can see here, that's the Tata box. And this whole thing is the promoter region. And so it's a section of the DNA before the gene that it allows for the connection of a generalized transcription factor, which signals to the RNA polymerase to bind and make RNA or to not do that. And so DNA binding proteins called transcription factors. So different transcription factors can be enhancers or silencers. So you can see up here, enhancers or silencers are going to regulate how much transcription is happening and how much RNA, messenger RNA is being made. And so activators will attach onto the enhancers, and they're going to speed up the rate of transcription of the gene. Whereas a repressor is going to attach onto a silencer, and that's going to make the gene not work or reduce its functionality. So when messenger RNA is made, it goes through a post-transcriptional modification process where a poly-A tail is added on to the messenger RNA. And this is a sequence of 100 to 200 adenine bases. And as the messenger RNA ages, the poly-A tail degrades, which reduces the translation of that messenger RNA. The lifespan of the messenger RNA and the poly-A tail can be influenced by hormones and other chemical signals. So high estrogen levels, for example, cause the messenger RNA that produces vitellogenin for making egg yolk to persist 3.5 times longer than when estrogen levels are low. And genome is all DNA, so all of it, coding and non-coding. So when we're thinking about our genome, we have coding genes, we have non-coding genes, we have mitochondrial genes, we have chloroplast genes. And then the transcriptome is going to be where RNA is made. And so that could be messenger RNA, could be non-coding RNA as well, like transfer RNA or ribosomal RNA. And so the transcriptome is going to be smaller than the genome. And then the proteome is going to be even smaller, and that's the enzymes or structural or functional proteins, non-structural proteins, all the proteins that are made in our body. And so the transcriptome is going to be bigger than the proteome. And then lastly is going to be the metabolome, but we don't need to worry about this. And so this is a modified version, like this is a piece of messenger RNA that has gone through transcriptional modification. So it has a 5' cap and a poly-A tail. And the poly-A tail, as I said over here, is going to influence how long it can translate for. And the 5' cap protects this end of the messenger RNA. So epigenesis is multicellular organisms have developed from undifferentiated cells, and differentiation is achieved by activating and silencing certain genes. We've talked about how a caterpillar has the same DNA as a butterfly, but they look very different. And so certain genes have been activated and certain ones have been silenced and result in the differences that we see. So this is achieved through chemical modifications to the DNA, and those are called epigenetic tags. And the DNA-based sequences are not changed, but what genes are active will be changed. So in the situation of methylation, methyl groups replace hydrogen on a base. So this is usually happening on cytosine, but could also happen on adenine. And methylation of cytosine in a promoter region represses transcription. So that means that messenger RNA will not be made. And nucleosome packing can be affected by the methylation of the amino acids, up or down by affecting the accessibility by transcription factors. So essentially, it's meaning that gene is coiled up tighter, and as a byproduct of that, transcription factors can or cannot access that promoter region, and so it will up or down regulate the production of that messenger RNA. And so here we have acetylation, which it's similar to methylation. And so when acetylation happens, typically the DNA becomes more loosely packed, and so will upregulate the transcription of that gene. So epigenetics, an organism's all of their epigenetic tags is called the epigenome, and the environment can affect the tags, which can be passed on through mitosis and therefore conserved throughout the lifetime of that organism. And so they've done twin studies on people who've gone to space, and one twin went to space and the other stayed on Earth, and how that affected that exposure to radiation, that training, that physical fitness, how all of those things have affected the epigenetics of the identical twin, because they started out with the same genome. Most, but not necessarily all, tags will be removed in the process of meiosis and fertilization. This is called reprogramming. And so all the epigenetic tags are mostly wiped off. Transgenerational epigenetic inheritance is transmission of epigenetic tags to the next generation. And that can be affected by environmental factors that affected the parents. And so what they have seen is, for example, females who got pregnant in starvation situations, the genetics, what genes are active in their offspring. If the fetus survived but was going through a very tight food situation, their genes will be more programmed to cause them to eat more when they're born. Another example is here that air pollution decreases methylation, which will increase immune response. And so as a byproduct of that, because you have a lot more inflammation, you have a higher risk of heart disease and asthma. But it's also problematic for pregnant women because the fetus can be affected, but it is possible to reverse those tags. And so, for example, as a smoker, you might end up with methylation occurring or on your cells related to smoking. But if you stop smoking, then that can reverse some of that process. So epigenetic tags are an example of how environmental factors can affect gene expression. If lactose, for example, is not present in the if lactose is not present, the repressor, that's this thing here, binds to the operator. And this whole thing is called a lac operon. So it's going to make lactase, sorry, which is the enzyme that will break down lactose. And there's a bunch of different lactase enzymes. And so this process will produce all of those lactase enzymes, which is why this is called a lac operon. So if the repressor is bound to the operator, and this is the whole promoter region here, then RNA polymerase cannot get through there, and it is not going to produce lactase. But when lactose is present, the repressor is deactivated and lactase would be made. Hormones can also play a role. So estrogen binds to estrogen receptors in the nucleus. And when estrogen receptors are activated, they trigger the production of progesterone receptors. So therefore, estrogen makes the endometrium of female's uterus more receptive to progesterone, which signals for those cells to grow more. So to summarize, messenger RNA is needed for gene expression. As messenger RNA ages, it will produce less proteins, and the poly A tail can influence this. Gene expression can be influenced by chemical signaling in the body. So for example, the lac operon, or the environment with epigenetic tags. And tags can be acetylation, methylation, or phosphorylation, which will have different effects up or down, regulating the transcription of the gene by affecting how the DNA is coiled around histones. So you should know the vocabulary. What is a silencer? What's an enhancer? What's a promoter region? And what's an operator? All right, that's it for this video. Thank you very much for watching.